आज हम रिकॉम्बिनेंट डीएनए टेक्नोलॉजी में जनरल इंट्रोडक्शन के बाद अपना सेकंड टॉपिक देखेंगे एंड द सेकंड टॉपिक इज अबाउट रेस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम्स। The restriction endonuclease enzyme. Some workers says that only use the restriction enzymes and so on. But there is a complete name is restriction endonucleases. First of all, we will see why they are called so. You know, nucleases which degrade the nucleic acid, are called nucleases, and this group is the endonucleases. We know that there are two groups, endonucleases and the exonucleases. Exonucleases may cut in DNA from the end point, means like from there, but endonuclease may cut within the DNA and cut both the strands of DNA. So we can say that endonuclease cut within the DNA. And why it is called a restriction endonuclease? The reason is that we know when in bacteria is infected with a bacteriophage and bacteriophage inserts DNA in the bacterial cell, the bacterial DNA secrete restriction endonuclease which choke up the foreign DNA. When we observed it and discovered the restriction endonuclease which restrict the growth of foreign DNA into the bacterial cell. Now we will see with some examples that name some restriction endonucleases. The number one is the most important, is the EcoR1. EcoR1. About the nomenclature of restriction endonuclease, it is clear that it was isolated from the bacteria Escherichia coli RY13 strain. So the first capital letter denotes the genus, second to small letter denotes the species, next R represent the type of strain, and first represent the sequence of isolation of that restriction endonuclease enzyme. So when we see at the large scale the types of restriction endonucleases, we can see that here there is the type 1 restriction endonuclease, type 2 restriction endonuclease, and type 3 restriction endonuclease. And in the RDT we use only restriction endonuclease type 2. Why? The reason is major that it cut within the recognition site. Cut within the recognition site, but that cuts around 1000 base pair. It cuts approximate 24 to 26 base pair above. So it is that is not useful for our. And both these need ATP for their activity, while type 2 don't need ATP for their activity. So these are the major differences between three groups of restriction endonuclease. Now we see that the EcoR1 needs an recognition site. Now we will see about the recognition site. Recognition sites are base pairs on DNA, either 4 to 6 base pair mostly. So we see here for the EcoR1 in the DNA, the recognition site is GAA GATTC. And important thing is that this is an palindrome. This is an palindrome. Palindrome किसे कहते हैं? वो सीक्वेंस जिसको अगर आप 5 प्राइम टू 3 प्राइम पढ़े, जैसे ये 5 प्राइम टू 3 प्राइम हम अगर इस स्टेट में पढ़ रहे हैं तो हम क्या पढ़ रहे हैं? GATTC. इसको भी अगर 5 प्राइम टू 3 प्राइम हम प्रोनाउंस करें तो GATTC प्रोनाउंस होता है. और कट ये जो लगाता है, ये लगाता है G और A के बीच में. एंड द रिजल्टेंट जो हमें डीएनए मिलेगा वो कुछ इस तरीके से मिलता है जाके. और इधर से तो these not bonded overhanging bases are called sticky ends. Sticky ends. It is most important to join the foreign DNA to this cutted edge. The edge should be sticky end, that knot should be blunt end. How blunt end are produced is also very important for us. And we see that like many enzymes, that is the enzyme HPA first. There is another enzyme HPA first, isolated from the Haemophilus parainfluenzae. Its recognition site is GTTAAC. Palindrome बनाइए GTTAAC. It cut between T and A. So it produces blunt end, that is not sticky end, because every base pair is bonded to each other. So these are blunt ends. For blunt, to convert blunt into the sticky end, we use adapters, linkers. They are the further topics in our syllabus. Next one is about the first restricted endonuclease that was discovered. It is very important for us that it was Haemophilus influenzae bacteria, a hint second. The first restricted endonuclease was hint second from the bacteria Haemophilus influenzae. तो आपको यहां देखना होगा कि देखिए जो इको R1 है या HPA फर्स्ट है या आपने हिंट सेकंड देखा, ये टाइप है एंजाइम के. दीज आर प्रोटीन्स मींस एंजाइम्स, एंड दीज आर क्लोनिंग साइट, रेस्ट्रिक्शन साइट, रिकॉग्निशन साइट फॉर दैट एंजाइम वेयर दे मेक कट. So we use restriction endonuclease to cut the DNA of our experiment. So they are also called molecular scissors. They are called molecular scissors. So thank you. It was everything about including the syllabus about the restriction endonuclease, their nomenclature, their classification, everything. The short lecture about it. Thank you.